Make sure your volume's up. Yeah. Oh, I thought this was me. Oh. Hey, let me start over. So this is the Peggy Carter Travel Award. We have 3 awards for early career scientists to go to the National ASM meeting, microbe meeting per year. Someone's talking.
Texting them. They weren't answering their phone, that the sound was off.
Sorry. Yes, Toby. We just turned the sound back on.
Thank you.
Thank you. So the award application form is online. So go to our website and you can either nominate yourself as an earlier career faculty, or you can nominate someone in your lab, either a postdoc or a clinical technologist. Unfortunately, students are not eligible. And in order to apply for the travel awards, you have to be a ASM members as well as a EPAASM members. And so here are some of our upcoming events. We have our 800th monthly meeting next month. We have Dr. Esther Babedi coming from Sloan Kettering. And then we have Dr. Olson in March. Kim Orth, who is our ASM Distinguished Lecturer in April. And of course, we have our student chapter organized meeting, as well as our education committee meeting in May and June. So without further ado, Kyle is going to introduce our speaker.
Great, thanks, Vincent. Very excited to see so many people here today. This is a great return and a fantastic venue. Thank you, Michelle, for organizing here at Drexel. So it is my pleasure to introduce Dr. Jason Carline. He is professor of microbiology and immunology at the Virginia Commonwealth University School of Medicine. His laboratory studies arthropod-transmitted obligate intracellular bacteria, defining the mechanisms by which these pathogens co-opt the host cell during infection. Dr. Carline's multidisciplinary approach to dissecting host pathogen interactions seeks to reveal targets for novel ways to prevent infection and also treat disease. The 2 main pathogens of focus in his lab include Orientia susu-gamushi, the cause of scrub typhus, and the topic of today's lecture, Anaplasma phagocytophilum, the cause of granulocytic anaplasmosis. Jason's contributions to the field are significant. He has over 79 publications, including a number of editorial spotlights, and he's been invited to give lectures on the national and international stage at many venues. But his contributions also include generosity of his time, serving as past president for the American Society for Rickettsiology. He's on the editorial board for a number of journals. He's had a number of years served as a member of the bacterial pathogenesis study section. And he's mentored students at all levels of learning, including 14 doctoral students and 18 postdocs. Dr. Carline's hard work has been recognized with continuous funding of his primary Anaplasma R01 for over 20 years at this point. Election into the American Academy of Microbiology in 2019, and a recent recognition of his achievements by colleagues in being awarded an R37 Merit Award to continue his research for years to come. Please join me in welcoming Dr. Carline as he shares a story of the expert cell biologist, Anaplasma phagocytophilum. You good? Thanks. Just need to share.
Okay, I think we're good. Can you hear me in the back? Okay. And I hope that everyone can hear me online. Okay. I guess you'll be, Shell will give you a call if there's a problem. Okay. Well, thanks everyone for coming. Kyle, thank you so much for hosting me. It's a real pleasure to be here, especially being hosted by a former student. Seeing as I'm the 799th speaker in this seminar series, it's a real honor to be part of such a long-standing tradition. Now, as the title of my talk indicates, the pathogen that I'm going to be talking about today, Anaplasma phagocytophilum, causes a tick-transmitted disease. And as I'm sure you're all well aware, tick-transmitted diseases are a significant health concern here in the United States and worldwide. But don't just take my word for it. Let's listen to this announcement from Terry Crews.
Remember fun, right? Nothing. This is Atlanta's awful guy. We get bored and all we think about is Lyme disease. Think about all tick-borne diseases, but mostly malaria, fever.
Okay, so as you can see, anaplasmosis ranks second on Terry Crews's list of concerning tick-borne illnesses. Now, coincidentally, this can't get enough of this video. Incidentally, granulocytic anaplasmosis is the second most common tick-borne disease in the United States behind Lyme disease. This disease can also be transmitted perinatally and by blood transfusion. And following inoculation, there's an incubation period of about 7 to 10 days, after which the disease presents as an acute febrile illness associated with a number of non-specific symptoms. Unlike Lyme disease, there's no rash. So unless the patient realizes that they've been fed upon by a tick, or the clinician ascertains that the patient could be at risk for a tick-borne illness, this disease can be difficult to diagnose, and delay in diagnosis carries the risk of progressing to more severe disease, which is characterized by disseminated intravascular coagulation like coagulopathies, hemorrhagic manifestations, renal failure, muscle loss, and toxic shock like syndrome. This also poses a risk for susceptibility to fatal opportunistic infections. 36% of patients that develop symptoms require hospitalization. 7% of them end up in the ICU. Now, in 2000, this disease became reportable to the CDC. And since that time, you can see that the number of cases reported annually has increased dramatically. But what's important to keep in mind here is that this is passive reporting. There's been a couple of active surveillance studies, and they have indicated that the true incidence for granulocytic anaplasmosis could be as much as 50-fold greater in this country. The majority of reported cases occur in the Northeast and upper Midwest, and in parts of these endemic areas, as much as 36% of the population has been exposed. So this is a zoonotic disease that naturally cycles between Ixodes ticks and a number of small mammals. Ixodes ticks are the same ticks that transmit Lyme disease. And strains that that are reservoired by the white-footed mouse, the gray squirrel, and chipmunks are associated with being able to cause disease in humans, dogs, and equines when accidentally vectored to them. Now, in their reservoir host and in accidental host, anaplasma exhibits this unusual tropism for neutrophils. Now, shown here in the scanning electron micrograph, you see several anaplasma bacteria bound to the surface. This is an image of a blood smear taken from a granulocytic anaplasmosis human patient. So here you have the nucleus of the neutrophil, and here's this vacuolar inclusion harboring numerous stained anaplasma organisms. So anaplasma is an obligate intracellular bacterium. Shown here is an electron micrograph. We can get a better view of this vacuole. This is the promyelocytic HL60 cell. Here we have the nucleus, and there are numerous vacuoles, each harboring anaplasma phagocytophilum organisms. Now, what I hope you can appreciate by looking at this electron micrograph is that there are two distinct forms, one which is more electron dense or darker than the other. And these play distinct roles in the anaplasma biphasic developmental cycle. So the more electron dense form is referred to as the dense cord form, or the DC for short. That's the infectious form. It binds to the cell surface, induces its uptake into a host cell derived vacuole. Within a few hours, it converts into the more electron lucent reticulate cell form. This is the replicative form. It's going to divide by binary fission, filling the vacuole with bacteria. The vacuole is going to expand to accommodate the growing intravacuolar bacterial population. And in tissue cell culture, after about 24 to 28 hours, anaplasma is going to convert back to the dense cord form, and between 28 and 32 hours, it's going to exit to disseminate and propagate new infections. So my talk's going to be broken into two separate parts. The first part is going to focus on this phase of infection, the binding and entry, and the second part is going to focus on the phase where anaplasma replicates and then exits the cell. So here we are in the first part. I'm going to tell you the punchline first, and that's anaplasma uses multiple adhesins that work together to facilitate its entry. So it's important to set the stage, to go back in history a bit to what stands as a seminal paper in the field. This came out of Jesse Goodman's group when he was at the University of Minnesota. And this was before anaplasma was even phylogenetically classified as an anaplasma species. It was originally thought to be an Ehrlichia species, and it was called the agent of granulocytic anaplasmosis. And in this paper, the Goodman group identified this selectin ligand called PSGL1 as a receptor for anaplasma. Now, PSGL1 is presented on the surface of neutrophils and other leukocytes, and it normally plays a role in interacting with selectin ligands on endothelial cells, and that's what allows neutrophils to roll along endotheliums and ultimately arrest at sites of inflammation. All of these branching structures on PSGL1 are glycans, and work that I was involved in as a postdoc helped determine that anaplasma binds to two particular sugars in this one glycan called Sialo Lewis X that's presented on the N terminus of PSGL1, as well as this stretch of amino acids in the PSGL1 amino terminus. So that sets the stage for this model that I am going to gradually fill in the information for as I progress through the talk. So what we're looking at, this is an anaplasma dense core surface with its adhesins interacting with SLEX capped PSGL1, and this is the neutrophil surface. So what we know from Goodman's work and our work is that anaplasma is interacting with SLEX capped PSGL1. From our work and others, we are aware that there's this signaling cascade that involves spleen tyrosine kinase, or SYK for short, and then these downstream events that are associated with cytoskeletal reorganization and bacterial uptake. So we were interested in trying to identify what this adhesion is. But work by other groups has shown that anaplasma enters at lipid rafts. Now, PSGL1 does not normally localize to lipid rafts unless it's activated to do so. So that led us to believe that perhaps there are other receptors that are localized in lipid rafts, and we set out to identify what those are. Now, this was not a particularly easy undertaking because the genetic tools for anaplasma remain a bit limited, and they were certainly limited at the time that I started this work. The other problem is that even if anaplasma was highly tractable, it's an obligate intracellular, and it has to require these interactions that allow it to invade in order to survive. So that was going to make it really difficult to use genetics anyway to try to identify mutants involved in invasion. So we had to take a bit of a brute force approach. So what we did is we took a biotinylating agent to label the surface proteins on anaplasma. We then lysed those cells and used streptavidin affinity chromatography to recover them, and we identified those by mass spec. We then applied bioinformatics to just look at proteins annotated as outer membrane proteins, or perhaps it had hydrophobic domains that could potentially serve as anchoring these proteins to the anaplasma surface. From there, we took our list and we did transcriptional profiling on them, focusing on those that are upregulated in either the infectious dense core form at the time that it binds to host cells, or when anaplasma transitions to the dense core form and exits during the infection cycle. We further focused on surface protein candidates that are upregulated during the tick transmission blood meal. So when the tick transmits anaplasma into a mammalian host, we then express these candidates in recombinant form and screen them with sera from human or canine anaplasmosis patients to see if they're expressed during infection in vivo. We used trypsin to see if we could remove these candidates from the surface of intact anaplasma. And from there, we took our candidates and expressed them in recombinant form and assessed for their ability to bind to host cell surfaces. And we did this using flow cytometry. From there, we generated N and C terminal truncation mutants to see if we could remove domains that were important for binding to the host cell surface. We then took these proteins and assessed their ability to competitively antagonize anaplasma infection of host cells. And together, this approach allowed us to whittle down to regions of roughly 50 amino acids that contained the putative binding domains of these adhesins. We then took overlapping peptides corresponding to those regions and raised anti-sera against each of those and assessed for those anti-sera's ability to inhibit infection. And using this approach over a number of years and a number of papers, we arrived at 3 proteins that are involved in anaplasma infection. They satisfy all the criteria that I just listed on the previous slide. And using that reductionist approach, we're able to whittle down the binding domains of each of these to these 12 to 15 amino acid stretches. Now, this is just some representative data showing that if we target any one of these proteins, in this case, we're targeting this protein called APA, its binding domain, we can reduce anaplasma infection of host cells in vitro. If we use anti-sera targeting 2 at the same time, we can get more effective blocking, and targeting all 3 gives us the best blocking. So now if we return to this model in data I'm skipping over, we were able to identify that one of those 3 candidates, outer membrane protein A, does in fact interact with that SLEX cap. but we still didn't know what was the targets of AS14 or APA were. So we started with AS14. So we knew that this protein is critical for anaplasma to enter host cells, and specifically it requires its C-terminal most 12 amino acids to do so. So we partnered with this company called Hybridgenics. They performed a yeast-2 hybrid screen using AS14 as bait and probed a human leukocyte expression library. And one of the interesting hits was protein disulfide isomerase. So as you know, PDI normally or typically plays a role in the ER where it functions as both an oxidase and a reductase and is key in quality control for protein folding. But PDI will also translocate to the cell surface where it associates with lipid rafts. If you recall, anaplasma binds at lipid rafts and enters as well. At the cell surface, PDI functions exclusively as a thiol reductase. And we were also encouraged by this hit because it's been previously shown that viruses such as HIV and dengue, as well as the obligate intracellular bacterium Chlamydia trachomatis, also require PDI for entering host cells. So we first wanted to validate this interaction. To do this, we took host cells and we transfected them to express either flag tagged PDI or both flag tagged PDI and GFP tagged AS14. We then used a flag affinity agarose to pull down PDI from these lysates. And we assessed for the ability for PDI to co-precipitate GFP tagged AS14. And we see that, in fact, it can do that. Importantly, it cannot pull down GFP alone, and it cannot pull down GFP tagged AS14 if it's lacking those final 12 amino acids that constitute the binding domain required for infection. We're able to extend this to anaplasmal organisms. So in this case, we're just, we can use PDI to pull down AS14 from the anaplasma surface. If we use siRNA to reduce levels of PDI in host cells, this has no effect on the ability of anaplasma to bind to the cell surface, but it reduces the percentage of infected cells and the overall bacterial load by about 50%. In agreement with this, if we take conditional knockout mice that lack PDI expression only in dendritic cells and neutrophils, and if you recall, anaplasma infects neutrophils, we are able to reduce infection on the peak days by about 50%. Now, in this slide, we're looking at the importance of PDI's enzymatic activity to infection. So in this case, we took this monoclonal antibody called BD34. This binds to the catalytic domain of PDI and catalytically inactivates it. If you treat host cells with BD34, you're able to reduce anaplasma infection of those cells by about 50%. And now if you repeat that in the presence of this membrane impermeable reducing agent called TSEP, that is able to reverse the ability of anaplasma to infect. In essence, it rescues it. So what this is telling us is that reduction or PDI's enzymatic activity is critical for infection. So when we next repeated this, we treated host cells with BD34, and this time we reduced only the host cell surface, and that was not able to rescue infection. But if you repeat it and you treat the bacterial cell surface with TSEP, that is able to rescue infection. So that's telling us that PDI is reducing disulfide bonds on the anaplasma surface, and that's what's important for anaplasma to infect. We took this one step further, where we took host cells or anaplasma and we incubated them with antibodies against either the AS14, APA, or AMPA binding domains. So antibody against the AS14 binding domain inhibits infection. TSEP is able to rescue that. But antibody against APA and AMPA also inhibits infection, and TSEP is not able to rescue that. So collectively, what this data is telling us is that AS14's interaction with PDI allows PDI to reduce disulfide bonds on the anaplasma surface, and that is critical for infection. And that's what's shown in this part of the model here. Now, in the next part, we've focused on APA. So like AS14, APA is also important for infection. The APA binding domain lies in this N-terminal domain that's surface exposed, and specifically it's amino acids 9 to 21. So our partnering with Hybridgenics again, their screen identified CD13 as an interesting putative hit. And CD13, we're excited about this hit because like PDI, it also localizes to lipid rafts. It plays a role in cleaving peptides at the cell surface, but importantly, it also elicits a number of signaling events that culminates in phagocytosis and receptor-mediated endocytosis. So when PDI is bound by a virus and a number of different viruses, when CD13 is bound by a virus, a number of different viruses bind to CD13 for entry, or if you use this monoclonal antibody called 452, either of those binding events promotes CD13 dimerization, and then that leads to this series of signaling events, which I want to point out involves SYK. That's the same kinase that is also activated by anaplasma binding to PSGL1 and is important for infection. And a number of these downstream events have also been shown to be associated with anaplasma entry. Now, the yeast-2 hybrid screen suggested that the C-terminal most region of CD13 is what APA binds to, and this is the same region that's bound by a number of different viruses. So the first thing we did is validate this interaction. In this case, we're expressing either the N-terminal region of APA, the first 87 amino acids, or that region lacking residues 1 through 21, and that includes the binding domain. So if we couple CD13 to beads, and then we can pull down GFP tagged APA, but not APA if it lacks the binding domain. We can also use these beads to pull down APA from anaplasma organisms, both as a monomer and a multimer. We presume this is a dimer, but we haven't done proteomics to validate that that's the case. Now, HEC cells do not normally express CD13, but if we transfect them to express it and we validate that CD13 is in fact on the cell surface by flow cytometry, and we incubate those with anaplasma, we see a 40% increase in infection of these cells relative to controls. Conversely, if you take human neutrophils and you treat those with CD13 antibody, you're able to reduce anaplasma infection of those by about 40%. You take CD13 knockout mice, the ability of anaplasma to productively infect these mice is reduced by about 40%. If you take an antibody that is specific for the APA binding domain and you incubate that with host cells, that inhibits anaplasma's ability to infect these cells by about 40%. And then that can be rescued if you then add monoclonal antibody 452. Now, if you recall in the previous slide, that's the antibody that will bind to CD13 and then elicit signaling events through this. So that suggests that APA is binding to CD13 and eliciting signaling events associated with anaplasma infection. And we see that this is in fact the case. So if you add anaplasma to host cells, beginning as early as 5 minutes, you get this elicitation of SYK phosphorylation, and this increases throughout the next 4 hours. And this has been previously published by us and other groups. But now if you do this in the presence of the APA domain blocking antibody, you're able to considerably reduce that. So now if we return to this model, coincident with these other binding events, APA is binding to CD13. We know minimally that this involves SYK, and we're investigating if these other events as well are elicited by this specific interaction. Now, at this point, we knew that AMP-A, AS14, and APA were all involved in anaplasma's ability to infect host cells, but we wanted to assess the relevance in vivo. So to do this, we looked specifically at whether we could immunize mice against the binding domains of each of these adhesins. So we conjugated these to keyhole limpet hemocyanin to increase their immunogenicity. We immunized mice, boosted them twice, and then 2 weeks after the second boost, we inoculated with anaplasma and then monitored the bacterial blood load over the next 4 weeks. Now, it's important to point out when we did our confirmatory ELISAs, we found that we elicited humoral immune responses against the AS14 and APE binding domains, but not the AMP binding domain. So even though that this was conjugated to KLH, this peptide was not immunogenic. And so when we're looking at data in the next couple of slides, it was only associated with the humoral immune response against AS14 and APE. So these are our non-immunized controls, and typically what we see is a peak of infection around day 12 that then subsides. And we see the same thing when we in our adjuvant only or KLH plus adjuvant controls. When we look at our immunization group, we're able to see a pretty considerable reduction in the bacterial load. So we next wanted to assess if we immunize against the AS14 or APE binding domain alone. So again, here's our controls. Immunization against the AS14 domain reduces infection, as does immunization against the APE binding domain. So we're pretty encouraged by this. We now know that at least these two are involved in infection in vivo, and you can elicit a protective response by targeting them. And what we're still missing, though, is what this protein is that PDI reduces. So to try to hunt for this, we took anaplasma and we incubated them with recombinant PDI or recombinant mutant PDI that was catalytically inactivated. We just had alanine substitutions in the catalytic domain. And we incubated those either in the absence or the presence of this reagent MPB, which will bind to free thiol. So anything that PDI reduces will get bound by MPB, and that is linked to biotin, so those proteins get biotinylated. And we then, to validate that we successfully labeled these free thiols, we probed Western blotted lysates with streptavidin and HRP. So we repeated this 3 times, recovered the proteins, and performed mass spec. The hits varied a little bit in each of the 3 experiments, but there were 3 proteins that we got every time. And one of those was pretty exciting because nearly a decade prior, we had characterized this protein to be drastically upregulated in both mRNA and protein expression during the dense core phase. So if you look at this mRNA expression profile, this the transcripts for this protein are upregulated 70-fold when anaplasma is in this infectious form. It's also dramatically upregulated during the tick transmission blood meal, and it's expressed during infection in vivo. It multimerizes both in recombinant form and in native form with anaplasma. And so we performed our yeast-2 hybrid screen with hybridgenics using this protein, which we termed APE-B for anaplasma invasion protein B. And one of the most interesting hits that we got was CD18. Now, CD18, it's surface localized on leukocytes. It localizes to lipid rafts, and it associates with PDI. So we thought this was a pretty conspicuous hit. Importantly, CD18 dimerizes with CD11 to form the beta-2 integrin complex on the surface of leukocytes. Now, this beta-2 integrin complex is in this bent or inactive form until it receives signaling from the inside out that's going to then switch it to this active state that's capable of binding ligands. And we're excited when we look at the literature to see what is the signal that elicits that conformational change, and it's actually when PSGL1 gets bound. So normally, when PSGL1 engages its selectin ligand, that sends this inside-out signaling to change this conformation of the beta-2 integrin complex that allows it to better bind to its ligand. And this is just a more detailed view of that signaling. We'll just focus on what matters. This is elicited when PSGL1 gets bound. This is a SYK-dependent process. Both of these are involved. We know are anaplasma targets and elicits. These other downstream kinases also are known to get activated when anaplasma binds. And it's this signaling that then leads to this conformational change. And then when the high affinity complex binds its ligand, it again elicits SYK signaling. All of this we know is associated with anaplasma infection. So if we take anti-sera raised against the entirety of 8B, we're able to modestly reduce infection, and the blocking ability is synergistically increased when we include it in a cocktail targeting the other binding domains. And we're currently working to identify the binding domain of 8B. But if we take an antibody against CD18, we're able to reduce the bacterial load pretty significantly. And if we take host cells that don't normally express CD18, transfect them to express CD18, we're able to increase susceptibility to anaplasma infection by about 3-fold. So now if we return to this model, this is what we have built so far. We know that OMP-A and likely an unidentified adhesion are interacting with the S-Lex cap and N-terminus of PSGL1. This is associated with activating this SYK-dependent signaling cascade, which very likely is what activates the beta-2 integrin complex to switch from its low affinity to high affinity state. When it does that, it is well known that the high affinity version of beta-2 integrin associates directly with PDI. Concomitant with this, APA is binding to CD13, which elicits signaling that minimally involves SYK. AS14 is interacting with PDI, which then reduces the disulfide bond between 8B and either an other 8B protein or an unidentified protein. We don't know what that is yet. And that in turn promotes 8B binding to CD18. And collectively, we think that these are building a signaling platform that involves SYK in these downstream events that promote anaplasma entry. So that is the first part of the talk. And now we're going to focus on the intracellular phase. And for years, it's a number of researchers have shown that the anaplasma vacuole is capable of interfacing with a number of different membrane trafficking pathways. How it was able to do so has been unclear. It's also been a complete mystery as to how anaplasma is able to exit host cells. And the punchline for both of those questions is here. And that's because anaplasma resides in this organelle called the multivesicular body. And this work we just published, this came out in November. This was a big chunk of the dissertation work for a very talented student in the lab, Curtis Reed, who's going to be defending this spring. So just as a primer, multivesicular bodies are intermediate compartments in the endolysosomal system, and they begin to form shortly after the early endosome has formed. They form by this invagination of the limiting membrane of the early endosome, and there's a scission event that results in this invagination pinching off into the lumen of the early endosome. And so you get this formation of these intraluminal vesicles. Now, this process continues as the organelle matures along the endolysosomal pathway. And this is forms the multivesicular body, or an endosome that's full of intraluminal vesicles. This interacts with a number of different organelles. It forms membrane contacts with the endoplasmic reticulum. It fuses with autophagosomes. It receives vesicular traffic from the trans-Golgi and ER. All of these cargo, when they get delivered to the multivesicular body, they're monoubiquitinated. So these multivesicular bodies are highly monoubiquitinated. From there, it matures into a late multivesicular body, which has one of two possible fates. It's either going to fuse with the lysosome, or it's going to mobilize and translocate to the cell surface, where it fuses with the plasma membrane, and it releases those intraluminal vesicles extracellularly, and those are called exosomes. Oh, and I should mention that these exosomes, these intraluminal vesicles, are highly enriched in cholesterol and sphingolipids. Okay, so a little bit more background on exosomes. So this is has been a burgeoning area of research really over the past decade. And exosomes are really critical cell-to-cell communicators. When they exit host cells, they're full of protein, lipids, and nucleic acids. They elicit a number of responses in the cells that take them up. It can change signaling cascades, induce inflammation, modulate immune responses, elicit a number of different metabolic processes, induce gene expression, and when they're taken up by neutrophils, can regulate neutrophil functions. Now, the role of exosomes in bacterial pathogenesis are poorly understood. Now, if we take that primer and look at the anaplasma vacuole, it has long been known to exhibit these endosomal characteristics. If you add BSA gold, which is a marker that will traffic through the endolysosomal pathway, it ends up in the anaplasma vacuole. This vacuole does not fuse with lysosomes. It forms membrane contacts with the endoplasmic reticulum. It fuses with autophagosomes. It is heavily decorated with monoubiquitinated proteins, and it acquires intraluminal vesicles from the trans-Golgi and ER. So that what we're looking at here is a 3D rendering of the anaplasma vacuole. This protein is an anaplasma secreted protein that labels the anaplasma vacuolar membrane. So we're looking at the vacuole from the outside. TGN46 is a marker from the trans-Golgi that gets delivered into the anaplasma vacuole. And these are DAPI-stained bacteria inside that vacuole that are closely associating with these TGN vesicles. And we see the same phenomenon with Durlin, which is a marker for ER and ER vesicles. All of these vesicles are highly enriched in sphingolipids and cholesterol, and the ability of anaplasma to parasitize these lipids has long been known to be critical for its ability to yield infectious progeny. And this is shown here. If we use siRNA to knock down levels of a particular Rab that's important for trans-Golgi delivery into the vacuole, we see that we drastically reduce infection. If we use this drug that alters cholesterol flux within the anaplasma vacuole, we're also able to drastically reduce infection. So there's a lot of similarities between the anaplasma vacuole and MBB, so we took a closer look at this. And looking at this by transmission electron microscopy, we see that the anaplasma vacuole, in addition to being full of bacteria, is full of intraluminal vesicles. We can see an example of one of them pinching into the lumen here. If we add BSA gold, it ends up on the membranes of these intraluminal vesicles. And LBPA, lysobisphosphatidic acid, this is exclusively present in MBBs, and we see it enriched in anaplasma vacuoles. So just to orient you, because we're going to look at a few confocal micrographs like this is a human neutrophil that's infected with anaplasma. Here's the nucleus, and these are anaplasma vacuoles. So here we've immunolabeled LBPA, so it's labeling LBPA within the anaplasma vacuole lumen and around the luminal face of the vacuolar membranes. P44 is an anaplasma outer membrane protein. And if we draw this line along this micrograph and we look at the immunosignal strength for both of these in this graph here, the bottom line is we see that there's a lot of co-localization of anaplasma signal with LBPA signal within the lumen of the vacuole. Now, the anaplasma vacuole we're showing here is acidic. So here you have two anaplasma vacuoles. D189 is a chemical that will fluoresce when it's in acidic pH of around pH 5. That's consistent with the pH of an MBB, and we see that the lumen of the anaplasma vacuole is in fact acidic. Conversely, if we use a dye that fluoresces in neutral pH, we see fluorescence in the cytoplasm outside these anaplasma vacuoles, but very little signal within. Now, the final thing that we wanted to do to prove our point that the anaplasma vacuole is hijacking MBB biogenesis was we looked for the presence of this sorting machinery that assembles on the MBB membranes called escorts. And this is called endosomal sorting complexes required for translocation. So basically, any cargo that's going to be delivered into an MBB is ubiquitinated, this protein called escort 0 will bind to that ubiquitinated protein. It recruits escort 1, which it recruits escort 2, and in turn that recruits escort 3. There's another escort independent pathway that involves this protein called ALEX. Both ALEX and the escort dependent pathway converge at escort 3, and escort 3 plays a role in this actual invagination and scissioning event that generates the intraluminal vesicle and carries that cargo into the lumen. So we just look for the presence of these proteins on the anaplasma vacuole, and long story short, we see components of the escort pathway, ALEX as well as this component of escort 3, where everything, where both pathways converge. Now, we did a number of different knockdown experiments where we looked for the contributions of these proteins to anaplasma infection, but the most important one involved when we knocked down expression of CHIMP4B, which is this escort 3 component, where everything converges and promotes the ILV formation. So this is our controls. Here you have a number of anaplasma vacuoles full of anaplasma bacteria, also full of intraluminal vesicles that are BSA gold positive. When we knock down CHIMP4B, we see that anaplasma gets in just fine. In fact, it gets in pretty well, but we don't see any replication. At the most, you see 2 anaplasma bacteria within the vacuole. And coincident with that, you see a drastic reduction in ILV formation inside. So when we look at the overall anaplasma load in their cells, we see that it's reduced by about 50%. And then we did a really exciting experiment where we looked for the relevance of the escort pathway to infectious progeny production. So in this experiment, we took media from either our control cells or our CHIMP4 knockdown cells, and we took that media at a time when anaplasma would have been released out into the media and would have been infectious. We then added that to naive cells, and then we looked for infection 24 hours later in the recipient cells. And what we see is this drastic reduction in infection, suggesting that the MBB pathway may be in fact important for anaplasma's ability to exit cells. So we looked at this by looking at the RAB, which is RAB27A, which is necessary for taking the MBB to the plasma membrane, where it will then associate with this protein called JCF1 and get it in what's called the exocytic zone. That then will, RAB27 then associates with the second protein called MUNC13.4, which then promotes snare-mediated fusion of these membranes and delivery of the ILVs out of the plasma membrane as exosomes. We see that the anaplasma vacuole is decorated with both of RAB27 and MUNC. And here we're looking at an anaplasma-infected cell by confocal microscopy. This is 16 successive focal planes going from the bottom to the top of the cell. And now we've compressed all of those into a one dimension, and we've color-coded the depth. So anything that is purple is at the bottom of the cell, and anything that is yellow is at the top. Now, if you look at this one anaplasma vacuole, we'll see it's at the bottom of the cell, and it has very little RAB27 associated with it. But here you have this larger anaplasma vacuole, and it has a lot of RAB27A associated with it. And what's interesting is when you look at the bacteria, they're not inside the vacuole, but they're around the periphery. So what has happened, if you look at this orthogonal view, is the when RAB27 associates with the vacuole, it lifts the it translocates the vacuole to the plasma membrane, where it releases the bacteria. So to prove our point, we use the small molecule inhibitor of RAB27A called NextInhib20. It binds to RAB27A, where RAB27A normally binds to JFC1. So it's going to prevent the ability of exosome release. Now, if we look at NextInhib treated cells, we see a modest reduction in infection no blockade in conversion to the infectious form. But what we do see is a drastic reduction in infectious progeny release. And conversely, what we see when we look at anaplasma vacuoles in these cells, we see that they are in fact retained. So that's telling us that RAB27A and the exosome release pathway is a key way by which anaplasma is exiting host cells. I'm almost done. I just want to tie all of this together. So we think that this is highly relevant for anaplasma's zoonotic cycle. When there's been very few studies done on the proteomics of exosomes, particularly from neutrophils, but in this one nice study where they were looking at the role of exosomes in asthma, they took neutrophils and stimulated them with LPS. And if you look at this Venn diagram, the punchline is that there is a dramatic change in the proteomic content when exposed to LPS. And there's a number of different proteins that are carried by these exosomes that regulate a number of different cellular processes. And in this study, what they looked at in particular in the context of asthma was the production of this, the increase of this protein called matrix metalloprotease 9. When my student Curtis did a literature search, he found a paper from years ago showing that anaplasma-infected neutrophils release more MMP9, and that is because anaplasma infection induces this persistent degranulation. So essentially, anaplasma is stimulating neutrophils to exocytose. It's using that pathway to get out. And a really neat paper that preceded our study came from Joe Pager's lab at the University of Maryland. He did this looking at tick salivary glands. The bottom line is that he found that tick salivary glands produce extracellular vesicles. These are, and when you look at the content of these are in fact exosomes. They benefit tick feeding. When they are incubated with immune cells, they modulate immune responses. And what was really exciting is he showed that anaplasma is able to modulate the content of exosomes. And if you inject anaplasma into a mouse in the presence of exosomes from tick salivary glands, anaplasma infects much better. So I think that this is really a new frontier to explore for the anaplasma field, but also intracellular pathogens in general. And as an example, we see that Ehrlichia and chlamydia, both of which are vacuole-adapted pathogens. If you look at their characteristics, they're highly similar to MBBs. This paper on brucella getting out using multivesicular bodies just came out this month. And a couple of papers from a few years ago showed that exosomes from mycobacterium and salmonella infected cells actually carry protective antigens, and you can use those exosomes to immunize mice, and that will induce protective immunity. So there's a lot of really interesting things to explore here. So just in a nutshell, that might be it, but that's the end. So I've shown that anaplasma uses a series of adhesins to promote its entry. It exploits the MVB pathway for promoting its growth within the vacuole and especially for releasing. So I really thank everyone for their attention. I want to thank my lab, my collaborators, and of course, my funding. So I'll take any questions. Thank you.
Very interesting. Thank you. So I have two questions. The first one is, so you have 3 adhesins, and it seems like 2 are in the wrap or intragenous protein back, and you knock down either immunize the mice with the 3 adhesin or you knock down individually. Do you have a sense of how those 3 together work together to mediate this invasion?
So what I think is it's safe to say is that all 3 of them culminate in activation of SYK, spleen tyrosine kinase. And we think synergistically that is promoting the cytoskeletal rearrangement. We are currently trying to tease this out to see if there's a hierarchy of one adhesion receptor interaction over the others. Is one more of a linchpin than the other in this process?
This seems like in the in vivo experiments, one seems to have a much greater effect.
Yes, so AS14 had a much stronger effect, and I think that makes sense because it's not if AS14 is utilizing PDI, which in turn we think is reducing APE-B, that the final adhesion that we characterized, which is critical for entry. And so of course, we're doing the experiments now where we're immunizing mice targeting specifically APE-B to see how robust of a protection we get.
And the second question, the NDP is also very exciting. So a lot of viruses also use NDP to fix the stuff. Right? So how does the bacteria know or regulate the exit, make sure that the infectious form before they get to the?
That's a great question. So I have a relatively new postdoc in my lab that came from a chlamydia genetics lab. And for his F32, he's proposing to do a genetic screen to try to identify what anaplasma, he thinks it's an effector that might be involved in mediating exit. So we don't know the answer to that yet, but hopefully a couple years we will.
So I was very interested in subsidiarity. So I agree, and these specific forms of chlamydia which are similar to what you have the second
Yes.
form with extremely reduced augmented protein. Did anybody look at the structure of molecules?
No, we have, well, first I'll say you're exactly right. If you were to look at transmission electron micrographs of, say, an anaplasma infected cell or a chlamydia infected cell, it would be very difficult to tell. They look morphologically very, very similar. And in terms of the crystal structure of these, the answer is no. But AS14 and APE-A exhibit a lot of disorder. And so I think if you did a co-crystal with them complex with their receptors, that might be a better way to go to try to understand their structure. But we have not done that yet.
And my second question is, do you ever look whether the exosomes in infected cells contain any of these APE-A?
Okay, so even though Curtis is pretty close to writing, he is very excited about this project and continues to do experiment after experiment. He has gotten really good at isolating pure exosomes, and we have done a couple of experiments. If you take anti-sera from an anaplasma infected animal, it recognizes a series of proteins. We have done some experiments to where it doesn't look like any of these outer membrane proteins are there, but there are certain secreted effectors that seem to be very abundant in these exosomes. And so anaplasma has a type 4 and a type 1. Interestingly, what we see is type 1 effectors. And I think the reason for that is because these effectors are released when anaplasma is inside the vacuole. They associate with the anaplasma vacuolar membrane. Now, if that's essentially an MBB and that membrane is pinching in to form ILVs or pre-exosomes, they're going to be enriched in those effectors. So what we are trying to do is isolate enough exosomes in enough abundance that we can do proteomics and lipidomics. And that's a big part of our R1 that gets reviewed in a month. So fingers crossed.
Hi, great talk. I have a question about your mouse model. A lot of the proteins you identified because they were upregulated during the blood meal. Do you do anything to prime the bacteria before injection to stimulate that response?
Not in that context. So what we inject are exclusively DCs. But admittedly, what we're missing there is any sort of synergy that occurs during the blood meal. The way that we laid this project out was to do syringe inoculation first, and a co-investigator on this project has a tick colony. He studies Lyme disease, and so we're going to repeat these very experiments. We're also, now looking at all of these interactions in the context of exosomes, like what role did the exosomes play in this as well?
Fantastic. I think that's all the questions we have. Jason, again, we really appreciate you coming here. Fantastic to host you for this talk, and fantastic talking. As this is the largest return to in-person, thanks for everybody that attended. See you next month for the 800th.
Thank you.